So the title of my talk today is Developing Optical Imaging Devices for Improved Intraoperative Breast Cancer Detection. First, I thought I'd give a bit of some information of how I actually got to this point and the research in the Perkins. So I actually grew up in Johannesburg in South Africa and then moved to Perth with my mum, my dad, and my brother, and we've got a little dog now, in 2010. And I went to school at All Saints College in Bull Creek, if anyone's around the area and knows it. And after finishing my studies at All Saints, I started study at UWA in 2016. Everyone kind of asks why or how I got into the field of biomedical engineering. It's quite like a niche field. I say it's a proof of genetics where my dad's an engineer, my mum's a doctor, so I split them 50-50 in the middle and did biomedical engineering to keep them both happy. So I started my PhD at Harry Perkins in 2021, and this is my sort of supervisor, Professor Brendan Kennedy, who's the head of the bioimaging lab here at the Perkins. Hopefully I should finish in 2025. Today I'll just give you a bit of a talk about what our lab has done. So it's important to recognize we're actually just one lab at the Perkins, and there's a lot of extra and other very diverse research. Breast cancer is the most commonly diagnosed cancer worldwide, and in an Australian context, 1 in 7 women are diagnosed with breast cancer in their lifetime. It used to be 1 in 8, but it's now kind of increased in 1 in 7, and that's quite clear with the sort of the increase in diagnosis by 21% in the last 10 years. So part of that is our tools for screening and detection are better, so we are catching more breast cancers, which is great. But there are also some lifestyle factors and that sort of thing that also contribute to that increase. The survival rates continue to increase with better clinical care, but there are still some issues, and that's because we have 2 main surgical treatment options that come with breast cancer. One is mastectomy, which is the removal of the entire breast as well as any sort of cancerous tissue. And we also have breast-conserving surgery, which is also called a lumpectomy, which aims to remove just the tumor and a margin of the surrounding healthy tissue while leaving most of the breast intact. So breast-conserving surgery is generally favored in early-stage breast cancers because the rates of survival is the same as a mastectomy. And it has several advantages with improved cosmetic outcomes, much shorter surgery and recovery times from that surgery, and better psychological impacts. But 20-30% of patients require an additional surgery when they opt for breast-conserving surgery. And this has increased physical, mental, and psychological burden when you have to keep going back to hospital for sort of your breast cancer treatment. And the reason that 20-30% of patients require that additional surgery is it's very challenging to ensure that you've removed all of the tumor during the first surgery. The reason we have this is when you're doing a breast-conserving surgery, what we try achieve is something called a negative margin, where you have some cancerous cells and that cancer is surrounded by healthy tissue. So you know it's completely enclosed and you've removed all the tumor. But there's also something called a positive margin, where they detect cancer on the edge of the excised specimen, which indicates that you may have left some tumor inside the surgical cavity, and therefore the patient has to go for an additional surgery to make sure that they've removed all the tumor. And the way that they actually detect if they've got a positive or negative margin and need to go for an additional surgery is by histopathology. So that's a current gold standard where the excised tissue will be sent to a pathologist who will analyze it in the days following the surgery. And if they see any reason for concern, then the patient goes in for an additional surgery. This is unfortunately quite a time-consuming process when the patient goes home and then they get retrospectively told, no, you have to come back for an additional surgery. So there's intraoperative margin assessment techniques that aims to look and see if they can detect that margin during the surgery, removing the need for the patient to go home and then come back for an additional surgery. So some key requirements of margin assessment techniques: they should be accurate, they should be able to sample the entire area that is required, and should be fast. We can't have the patient waiting in surgery for a long time, because that has its own additional risks when you have a long surgery, and preferably minimal operator training if we have a margin assessment technique. So a range has been developed, such as frozen section analysis, imprint cytology, specimen radiology, and optical coherence tomography. So these are all 4 techniques that have been developed to try, aim, and see if we can detect those tumor margins intraoperatively and save the patient from coming back. But each of these techniques has its own advantages and disadvantages. Some are not very accurate, some are very expensive, and some are very slow. So they don't necessarily meet all of the key requirements required for a very effective intraoperative margin assessment technique. So we've taken a slightly different approach at this, and we looked at what do surgeons routinely use to already detect cancer during the surgery. And that brings me to mechanical properties of tissue. So when doctors are doing the surgery, they use their sense of touch to identify the disease. And the reason is that because it's been increasingly well recognized that pathology and disease affects the mechanical properties of those tissue. And I'm sure if you've been to the doctor or physio and you've got like a bruise or some sort of inflammation, they'll touch it, and based on how it feels, they can make a diagnosis. And this made us realize that if you measure the stiffness of tissue, it can give us an insight into the onset and progression of disease, for example, sort of breast cancer. And I like to give this example. So this manual palpation technique where they use the sense of touch to identify disease dates back a long time to 1500 BC, and nothing's changed. So this scroll here is called Ebers Papyrus, and it was the first essentially medical textbook where in the ancient Egyptian times they had a scroll, and that scroll essentially had some details about how they would feel the breast for lumps to see if they could identify issues or disease. And not much has changed since then. But with the addition of technology, we do actually have a few additional ways to look at this. So that,
can I just ask
yes.
The difference in having your hands as against the rubber gloves for testing?
So the surgeons do wear rubber gloves, which reduce the sensitivity of being able to feel. Yeah. So I think we have done some initial studies, and they can't, with rubber gloves, detect tumors accurately if they're less than like a millimeter in size. Thank you. So elastrography is a group of imaging techniques that measure the mechanical properties of tissue. And this is a way to, instead of having sort of manual palpation by doctors and surgeons, can we provide maps of mechanical properties using imaging techniques? And this has been used in a number of clinical applications already. So ultrasound is like a very well developed technique, and ultrasound elastography is used in diagnosing liver fibrosis and a few other diseases. So the sensitivity and resolution of these depending on what imaging device you use. So magnetic resonance elastography is very good if you want to image the whole body and are not that concerned about seeing very small features. And then it goes from magnetic resonance elastography down to optical coherence elastography, where you can't measure very deep, but you can see very, very small features. So the choice of which one you choose depends on what application you're trying to solve. And what I'm going to be talking about today is optical coherence elastography, so that last one there, which is what we have developed and applied to margin assessment. So optical coherence elastography is based on an imaging technique called optical coherence tomography. So if you've ever been to the optometrist and you have to put your chin on the ledge and they scan the back of your eye, that's the same imaging technique that we are utilizing to develop this. So it was initially developed in about the 1970s, 1980s, but has been popularized since the 2000s as technology has developed. And to get elastography or stiffness images, we require 3 steps to do this. The first one is you apply a load. So if we have, let's say, a sample which is indicated in blue, we can go from an unloaded to a loaded state. We apply some sort of compression or palpation, much like a doctor or surgeon would do with their hands. It would apply a force or pressure. When we go from this unloaded to loaded state, we then track the deformation using the imaging device, almost creating a video of how the sample or tissue moves, going from our unloaded to our loaded state. And last, we use a mechanical model where we then reverse engineer based on the movement of the tissue, what parts are stiff and what parts are soft. And this can create images such as this, which is an image of a fibroedema, where all the blue regions indicate soft tissue and all the red regions indicate stiffer tissue. So we can start to see contrast between different tissue features based on its mechanical properties. And some benefits of this technique is it's very, very fast, and we can produce images in less than 5 minutes. We can get very, very high resolution images between 5 to 50 micrometers, and for reference, the width of a human hair is about 75 micrometers. So we can identify very small mechanical tissue features on the order of the size of a hair. And we can also produce 3-dimensional images on a depth of 1 to 2 millimeters, which is not very deep, but when you're just looking at the edge of those margins, that's all we need. So we are prioritizing seeing very, very small features over the larger imaging depth. So we developed this technology and did some studies ex vivo, where we took the tissue from the surgery and then scanned them in the lab. And we could scan regions of 5 by 5 millimeters, allowing us to do very rapid imaging. And what we showed is we could delineate malignant and healthy tissue. So this histopathology is what we got from the pathology lab, where the sort of darker purple regions show invasive lobular carcinoma, and the kind of pink regions are your surrounding sort of healthy tissue. And what we did is we processed that to its mechanical properties, which is shown in this last image in here, strain, where here we get these like circular structures, which you can then also see over there. So we can start to delineate the healthy tissue from the cancerous tissue based on its mechanical properties. We then developed an additional step, because right now we had something called a qualitative measurement, where we could only tell that something is stiffer than something else. It's like if I gave you 2 weights and you had to pick which one's heavier. You could tell me which one is heavier, but you couldn't tell me exactly how heavy that is unless you had a good reference. So we provided a reference by using a silicon layer, which we place on top of the tissue. And we've measured that silicon layer exactly. We know its mechanical properties. And by comparing its properties to the properties of the tissue, that's almost like if I gave you another weight and said, this is exactly 2 kilograms, then you can give me a fairly good estimate of how heavy the other ones are. So this allows us to produce the last image, which we call an elastogram, which shows the quantitative mechanical properties. And some benefits of doing this additional step is we get much higher contrast between the malignant and the healthy tissue. And also it allows us to compare tissue between patients or over the same patient at different times throughout their cancer journey. And we took this technology and we've done a diagnostic accuracy study on 83 patients and took 7 readers, which was some engineers, some clinicians, and some pathologists, and got a sensitivity and specificity of 93 and 96%. So that's a really encouraging start to say that our technology can actually be useful in helping diagnose and tell the difference between healthy and malignant tissue. But the surgeon said they would actually prefer to detect the cancer inside the surgical cavity. So what the current method uses and has been used in the past is we look at the tissue that's been excised from the patient, which is indicative of there might be some cancer left inside the patient. But if we can actually check the surgical cavity inside the patient themselves, that gives us a much more accurate representation of if you've actually still got cancer left in the patient. So that prompted us to develop a handheld imaging probe, which is shown over there. So that's the engineers like all the labels, because that's kind of the nerdy stuff that appeals to us. But this is what it looks like when we actually give it to a surgeon to be used in doing surgery. And so far, this probe has been used to scan about 80 patients here at Charles Gardner. We've also got Fiona Stanley, Royal Perth, and also Royal Melbourne as well. So there's a few sites that are being used to test this probe and its use during surgery. And this is just an example of its use from a scan that we took where we were able to see the blue regions, which was benign or healthy tissue, and see cancer by the red region. So this prompted then the surgeon to say, okay, we don't think we've removed all the tumor. I'm just going to take a little bit extra just to make sure, and hopefully the patient then doesn't have to come in for an additional surgery. So this has all been done by sort of Bright Lab and the lab I work with, but my work specifically aims to improve two things about this technique. The first one is to improve elasticity accuracy. So as we do all our measurements, there's a few assumptions about the tissue and its motion and movement that allow us to get those mechanical properties to guide the surgeon. But we recognize that when you have friction or complex tissue structures during surgery, it can reduce the accuracy of our measurement. So over the first year of my PhD, I developed an imaging protocol which allows us to improve the accuracy of our measurement. So this is a measurement before where we took this is a phantom that mimics sort of cancerous tissue, where we have down here a healthy region, and this little block there is a simulated stiff tumor. And when we get our elastogram image, you can't see much of a difference between our surrounding healthy tissue and the tumor. And after we applied the improved imaging protocol, we get a much clearer differentiation between the cancerous tumor, the cancerous tumor and the surrounding healthy tissue. So that allows us to be more confident that we're not missing sort of any tumor during surgery. Another one is to improve the clinical suitability. So currently we have to use that silicon layer to provide that reference and quantitative measurements. But that brings a few extra steps and complications where we have to teach the surgeon how to apply the layer, and also limits our ability to take the probe and do sort of endoscopic studies and even more difficult areas of the body to get to. So in my second PhD project, I implemented a way where we could combine this layer into the imaging system. So it's one fused device. And what we found is it increased the reliability of all our measurements because you remove that human judgment and human error from doing the measurement, and also provide us much more accurate measurements. So this is an example of a breast scan that we did where all of the sort of red regions indicate our regions of tumor. And afterwards, we get a much clearer delineation between the tumor and non-tumorous tissue. And that can allow the surgeon to be more accurate and precise when they're actually trying to remove the cancerous tissue. And that's kind of our work in the breast cancer, but we've also applied this in a few other sort of settings where this device that we've created, we've also applied it to imaging of cell mechanical properties. So rather than just looking at the tissue that comes out of the surgery or during the surgery, we've actually started to look at individual cancer cells as well to say, why do they actually get stiffer as cancer develops? And we've also taken this and developed what is called a mechanomicroscopy system, where we've integrated our imaging system with something called fluorescence imaging. So we can look at the activity of the cells. So why do the cells behave the way they do, and how does that link to mechanical properties? And the hope for that is we can then see the stiffer tissue, what parts of the cell are being activated or non-activated, and that can potentially lead to better treatments of cancer in the future. So there's a few different ones, and I've talked about quantitative microelastography today, which is all the images I've shown so far. But there's also been a few other developments where we've shown this technique, but we go, can we make this more accessible and also cheaper? Because the health system loves to see if we can get the same result for a cheaper device. So using that, we've developed something called OCT, optical palpation, which does the same thing, where it's meant to look at a stiff region and identify it based on its mechanical properties. And we've also developed something called stereoscopic optical palpation. So rather than using an OCT imaging device, which is quite a fancy, expensive imaging device. Stereoscopic optical palpation just uses 2 cameras. And it works almost on the principle of your eyes, where the ability to have depth perception is because you can have 2 eyes, and if you cover one, everything moves. And if you cover the other one, it moves the other way. And based on how much it moves with respect to one another, we can tell how far close something is. And we use the same technology that essentially your eyes do with cameras. And we can then therefore produce also maps of tumor and identify stiff regions in something that is a few $100s rather than many $1,000s. And this is particularly came out when we did COVID research during sort of 2020 and 2021. We realized that clinicians and specialists who were flying from Perth out into regional regions couldn't actually do that anymore. So we needed to provide a cheap and accessible device to doctors out there inside rural areas so they can do diagnoses by themselves rather than relying on the movement of specialists from key hospital centers. And that's pretty much all I've got today. So I'm just one of the members in the team, but you can see there's a pretty big research group led by our lab head, Professor Brendan Kennedy, and all of us are working on different aspects of the device and imaging technique to move it forward. I'd like to welcome any questions you might have.
So you're doing all this research here, which is all wonderful. How do you get all the surgeons on board and learning all of this so that all the surgeons that are doing these operations are all using your wonderful research?
Yeah, that's a very good question. So this started in collaboration with Professor Christabel Saunders, who's a breast surgeon, who's a part-time clinician and part-time researcher at UWA, and has since expanded to a bunch of other surgeons where we've approached surgeons at different hospitals and kind of shown them this technology and asked them if they'd be willing to learn and develop this device. So it does take a few engineers where we go in with them and teach them how to use the device. And there was also a startup company called OncoRes Medical, who are just actually down the road on Hampton Road, that are commercializing this device. So their aim is to make it more accessible and user-friendly so we can get it to different surgeons. And the aim is, after we've got sort of FDA approval and a few other sort of regulatory approvals, then it can start to be more widely adopted by surgeons rather than us having to approach them each time.
How do they take, when you come forward with these ideas, and does it take a long while for them to learn, and how do they learn? I mean, what do they practice on to learn how to operate these instruments?
It does take a while. We've kind of found we've also changed the way we do it as well, like not just the surgeons. As we develop the device, the way we do the measurements has changed. But we find that the surgeon on like the first or second go sort of needs a bit more practice. But we've developed like a fairly rigorous protocol to teach them how to use it. Sorry, what was the second part of the question?
How did they learn that? I mean, how did they learn to use it on tissue? Do you have tissue samples in the lab that they practice on?
Yeah, so we don't keep real sort of tissue around. We've developed something called tissue mimicking phantom. So it uses silicon, so the same material that is used to create prosthesis or breast implants, where we can essentially simulate a proper tissue, and we can say, okay, we have embedded a tumor, so can you find it? And we kind of train them and teach them how to use it sort of outside the surgery, then before going into the surgery, and then they develop how to use it. And we do have our engineers inside the surgery with them as well to try assist them during it until they feel more confident, and then they can do it by themselves.
So this is all trial now?
Yeah.
All these different hospitals are trialing, and how long do you do the trial?
I think the current trial using the handheld probe has been going for about 2 years, and the aim of that is to collect enough patients. So when we go to for regulatory approval to get the probe approved for use, you have to have demonstrated that you've used it in sufficient enough patients so that your data size is big enough to show, is it worth using, as anything new in surgery, it has to provide a significant benefit over what is currently there, because there's always risks of introducing new technologies and extending that surgical time. So we're hoping to have that sort of surgical accuracy study in the next year or so, and then can progress to the next stage of development.
So currently surgeons would be using it running alongside what they're already doing?
Yes,
and
yep.
Use it as an additional tool, not the only tool, until it gets all the approval?
Yeah, so the current protocol is we try integrate into their clinical workflow. We try interrupt as little as possible, where they will do the regular surgery, will then use the probe, and then have their clinical judgment based on our technique, as well as a few other identifying factors if they feel they need to take some more tissue. Yeah.
So this is actually in operation now with surgeons. So those surgeons that are using it, are they giving you the data? Are they part of the trial, or are they, okay?
Yeah, so they're part of the trial. So they're on our sort of research team. So anytime we do any sort of development and publication, they're always part of that team, which I think is really important. Specifically when we started developing the probe, we had something that we thought was really great. And as engineers, we said, this is brilliant. But then the surgeon came and said to us, oh, it's really heavy, it's not comfortable to hold. So it's all these practical aspects, which as engineers we hadn't really considered. Like the surgeon needs to hold it, it needs to be able to manipulate. And one of the other imaging devices we've got is we're developing it to be lightweight and wireless. So there's no cables. There's something in surgery called spaghetti syndrome, where the cables are running all over the place and it can be difficult to get around. So if we can develop something that has sort of minimal wires and is easy for the surgeon to use, it is the goal.
Can you see this probe being used for other general surgery, other areas other than breast?
Definitely. So we've also recently taken the technology to Poland as well. We've got a few Polish researchers who've looked at now prostate cancer. We've done a few sort of studies on skin as well. So we worked with Professor Fiona Wood looking at burn scars, because burn scars have the same thing. When you have scarring of the tissue, it stiffens, and that's actually how they grade your scarring during hospitals. They'll palpate it and feel how stiff it is. And we can actually track as your scar heals, it gets softer. And previously that has all been done qualitatively by clinicians, but they now have something that's quantitative, and they can actually put a number and track how somebody's healing. And we also thought that was very important, again, for, I think WA has 2 specialist burn centres, but nothing in the regional areas. But that's actually where most of the burn injuries happen. So we recognize this disconnect where people in regional areas actually require this technology, so they don't have to rely on either flying the patient here or the doctor to those regional areas to do what is actually regular diagnostics over time.